Swastiastu. Good morning, Doctor. We are from small group discussion B2. Today we will present our student project with the title PCR in Diagnosis of Infectious Diseases. Before we start our presentation, we will introduce ourselves. Next slide. The member's name and the student numbers can be seen on the slide. Okay, now we will start our presentation from the first chapter, Introduction. The background that will be presented by my friend.
Okay, my name is Nirajanari Putri. My last three digit of my student number is 067, and today I'm going to explain about the background of this student project. Infectious disease is exploring the world nowadays, especially in Indonesia. Infectious disease, based on the definition, an illness that exists by a pathogen or microorganism such as bacteria, fungi, virus, and parasites and can cause carrier. Some of the infectious diseases are contagious, therefore the spreadness of infectious disease need to be checked regularly and periodically. This is done to get the faster and accurate therapy, prevention, and target goal can build the effectiveness and efficiency. PCR or polymerase chain reaction is one of the methods to diagnose infectious disease, such as COVID-19, HIV, CMV, hepatitis B, and C. There are so many types of PCR with different uses and mechanisms to check the infectious diseases, and based on some research that has been done, PCR is one of the most effective methods to diagnose the infectious disease. Therefore, the author is interested to discuss the use of PCR in diagnosis of infectious disease. That's all my presentation. Thank you.
Okay, my name is Made Prema Pratnya Pesanti with 3 digits number of my, with the last 3 digits of my student number are 064. Now I will present the problem formulation of our paper. The first is, what are the definition and types of PCR? The next is, what are the definition, types, and inspection of infectious diseases? How is the mechanism of using PCR? What the use of PCR? And next, what are the advantages and disadvantages of using PCR? And the last, how is the future of PCR? Next, I will present the objectives of our paper. Is to know the definition and types of PCR, to know the definition, types, and inspection of infectious diseases, to know the mechanism of using PCR, to know the example of using PCR in everyday life, then to know the advantages and disadvantages of using PCR, and the last is to know the development of PCR in the future. Next slide. Now the benefits. There are 2 benefits, theoretical benefits and practical benefits. The theoretical benefits, this paper can be used as a source to find out about infectious diseases, PCR, and the role of PCR in diagnosing infectious diseases. This paper is expected to increase the knowledge about PCR, infectious diseases, and the role of PCR in diagnosing infectious diseases. Practical benefit. For readers, this paper can be used as a source of information and reference regarding PCR, infectious diseases, and the role of PCR in diagnosing infectious diseases. For writers, this paper writing can increase the experience, knowledge in writing scientific works. And the last, for academic institution, this paper can be used as a reference to develop PCR in diagnosing infectious diseases. That's all from me, and the next slide will be presented by my friend. Thank you.
Okay, hello everyone. My name is Putu Ari Nirajanari, and the last 3 digits of my student number are 049. I'm here to explain what is the definition of PCR. Okay, so PCR, or polymerase chain reaction, is a technique or method of DNA amplification that can amplify large amount of DNA segments up to millions of times in just a short of time. Because of that, PCR is also found to be the most widely used method in molecular biology because of its fast, cheap, and simple process. There are 3 stages and steps of the PCR. The first one is denaturation, and then the second one is annealing, and the last one is extension. Okay, that's all from me. So the next slide will be presented by my friend. Thank you.
Okay, so my name is Vyasa and my last three-digit name is 051. So I will explain a short explanation about history of PCR. So the beginning of the invention of the PCR technique was when the invention of recombinant DNA technology in 1980. And this discovery will be the beginning of the development of various technologies in the molecular field. For recombinant DNA technology, it actually requires a relatively large number of pure DNA samples, and to obtain it is often very difficult, especially if you have to be in contact with humans. And one possibility is if a small DNA sample can be amplified in vitro so that it can have a large stock of DNA. This natural DNA amplification system made the polymerase chain reaction or PCR technique developed and made it possible to produce pure DNA in large quantities. In the 1971, Clapp and his team wrote a journal consists the information about the technique that used enzymatic technology to replicate DNA templates. And in 1983, Mullis, that was working for Status Corporation, determined to develop the technique for DNA synthesis using short chain, which was derived initially from the PCR technique. They speculated that there is a method for amplifying DNA by duplicating the DNA with the enzyme DNA polymerase and in a repeatedly manner. He received some attention in the current year, and he was awarded a Nobel Prize in the chemistry. And the next slide will be presented with my friend.
Next slide, please. Hello, everyone. My name is Dinaria Sakura Kirani. The last three digits of my student number is 055. And here I will present to you types of PCR. The first one is their standard PCR, which are carried out one pair of primer that implies the targeted genomic sequence. The second type is reverse transcription PCR, which enable quantitative detections levels of RNA expressions by creating complementary DNA. The third type is real-time PCR, which is designed to carry out these reactions that comprise a thermal cycler for DNA amplifications. And the last type is RT-PCR or qPCR combined. Both RT-PCR and qPCR techniques are merged, and this combined techniques is called QRT-PCR or quantitative RT-PCR or RT-qPCR. The next slide will be presented by my friend.
Okay, I will continue my friend's presentation. My name is Kadek Bintang Putri, and my last three digits of my student number is 056. And I will explain to you about the definition of an infectious disease. Infection is a term that defines how the infectious agent enter and develop in the human body, whether it develop into a disease or not. The diagnostic technique of infection and disease will be able to change with the development of new diagnostic technique, which will be able to detect earlier signs of the action of the infection in the organism. It makes the limit between infection and disease are not always clear. Infectious disease is an illness from transmission of a specific infectious agent or its toxic product. It could be from infected person, animal, and reservoir to a vulnerable target. Infectious disease has 6 components. The first one is the agent or cause. The second one is reservoir of the disease. The third one is a portal of exit. The fourth one is transmission. The fifth, portal of entry. And the last one is host vulnerability. And the next slide will be presented in my friend. Thank you.
My name is Putu Ari Apsari with the last 3-digit number of my student number is 058. I will continue this presentation by explaining about type and inspection of infectious disease. Infectious disease can transmitted by insects or other animals and may get by consuming contaminated food or water and sometimes being exposed to organisms in environment. There are 4 pathogens or disease-causing infection, such as bacteria, fungi, parasitism, and virus. They have one common to cause infection and must invade by a host interacting with the host cell membrane cells. Some examples of infectious disease and their cause can be read on screen. Next, please. The next is inspection. Infectious disease can be exponentially transmitted among population in a relatively short period of time. Therefore, sensitive, specific, and rapid diagnosis testing plays a critical role in preventing the transmission of infectious disease. Nowadays, there are several ways to test infectious disease, such as the first, point of care testing or POCD, which combines simple, rapid, and exact on-site diagnosis platforms. Second, mass spectrometry-based approach to characterize respiratory and other viruses with fine molecular detail. And the last one, polymerase chain reaction or PCR, is a molecular biology technique used to multiply certain DNA fragments. That's all from me. Thank you. Next, please.
Okay, I will continue the presentation. My name is Kanya Amaris with the last three digits of my student number is 060. Now I will present about the mechanism of PCR test. The mechanism of how PCR works is based on the principle of DNA template, the sample DNA that contains the target sequence. At the beginning step of the reaction, high temperature is applied to the original double-stranded DNA molecule to separate the strands one from each other. There is a type of DNA polymerase enzyme that synthesizes new strands of DNA complementary to the target sequence. The first and most common is the use of PAQ DNA polymerase, whereas PFU DNA polymerase is used widely because of its higher fidelity when copying DNA. Next, please. The principle of PCR to amplify the fragment of DNA is by heating the DNA sample with high temperature until the DNA denatures and separates the DNA into two pieces of single-stranded DNA. Then PAQ polymerase enzyme synthesizes and builds two new strands of DNA using the original as a template. So the result of this process is the duplication of the original DNA. Each strand can create two new copies, and the cycle is repeated 20 to 40 times, resulting more than 1 million exact copies of the original DNA segment, like the pictures shown in the slide. This is all the presentation from me and will be continued by my friend. Thank you.
Okay, I will continue. My name is Putu Pranadea Vidasagara, and the last three digits of my student number are 059. And now I will explain about the using of PCR tests. First, as we know, PCR is a form of laboratory inspection that is test on cells, bacteria, or viruses to detect the presence of their genetic materials. Many areas of study, such as medical diagnosis, forensics investigation, even food analysis, have been applied PCR due to its versatility, specificity, and sensitivity. In food analysis application, PCR is used for the detection of ingredients of food products or different animal and fish species. Next, please. While in many scientific fields, PCR has some important roles which can be seen in this slide. Next, please. PCR is crucial for the diagnosis of microbial infections and epidemiological studies. PCR is the most sensitive test that can be used to detect herpes simplex virus, varicella zoster virus, human papillomavirus infection, cytomegalovirus infection, hepatitis C disease, and those that are currently spreading right now is for detection of the SARS-CoV-2 virus in COVID-19 patient. The AIDS virus and human immunodeficiency virus were also detected more quickly in just the first few weeks after infection if using quantitative PCR than standard ELISA tests. Other diagnosis uses including tests for infectious disease that caused by bacteria such as gonorrhea, chlamydia, Lyme disease, and pertussis, genetic disease, cancer, and other infectious diseases that are still constantly developing. That's all from me. Thank you.
Okay, I will continue the presentation. My name is Kadek Bintari. The last three digits of my student number is 063. Today, I will explain to you about the advantages of using PCR. The advantages of using PCR can be divided into two. The first is sensitivity of PCR. Based on the research, it was found that primers affect the sensitivity of PCR reaction. The second is specificity of PCR. A highly specific PCR will generate one and only one amplification product, that is the intended target sequence. That's all from me. The next slide will be presented by my friend. Thank you.
Hello, my name is Utami Utami, and my last digit number is 048. And here I'm going to explain about the disadvantages of using PCR. Despite being the gold standard, PCR has its own disadvantages. PCR can be inconsistent for detection. PCR has stressed to bring out false negative and false positive results for infection of COVID-19. The false negative results may occur because of the mutation in the primer and probe target in the SARS-CoV genome. RT-PCR depends on capturing and detecting the virus, so it has the potential to miss patients who recovered from the disease because the RT-PCR test only detects the active virus. For COVID-19, RT-PCR cannot provide any information on other diseases other than COVID-19. It can only tell if a patient is infected with a particular type of coronavirus. That's it for me, and the next slide will be presented by my friend.
Thank you. I will continue the presentation. My name is Gede Pramaditya Saputra, and the last three digits of my student numbers are 045. In this occasion, I would like to present the foreseeable future of PCR. PCR has been a great deal in the world of microbiology, biotechnology, and pathology. We have seen a lot of PCR innovations, especially when the pandemic of COVID-19 hits in the last 2 years. Some of those innovations are qPCR and RT-PCR. In the future, in order to achieve other innovations of PCR to enlarge its functions, PCR can be used with the help of other devices such as microfluidic devices for the analyzing process of immunomagnetic exosomal RNA and other functions. We could also see how PCR is combined with a smartphone that includes a fast response, user-friendly, and affordable instrument. The most important, PCR will have major improvement to prevent false positive and negative results with the combinations of more advanced products such as NGS. That's all from me. The next slide will be presented by my friend. Thank you.
Okay, let me introduce myself. My name is Anindya. The last digit of my student number is 067. I'm going to explain about the conclusion and advice of this student project. The conclusion of this student project, based on what have been discussed, this group can conclude that infectious disease can contagious, so PCR is needed because it can diagnose the infectious disease so the health workers and government can determine the effective therapy, prevention, treatment. There are several factors that need to consider for the success rate of the PCR process, such as temporal analysis of the two primary pairs, DNA concentration, and quality concentration of magnesium chloride, and etc. With the practice of using PCR that have that the future researchers can do the research appropriate to the target goal. This what needs to increase our addition knowledge about PCR. The advice first to the government. Next, please. Advice. First, for the government, they need to socialize more about the development of the PCR, especially in Indonesia. So, because COVID-19 is still one of the most infectious disease that spreads so rapidly and has many victims. Hopefully, that socialization can increase people's knowledge about PCR. Second, to the citizens, they need to have more literature about PCR and have more information selection. Need to be smart citizens because nowadays there is so many information of the internet about infectious disease and PCR. And the information does not always say the fact. So have to select information, especially using the scientific paper as the literature material. And the last one, for the future research, need to do the future research about this topic, especially to increase the effectiveness of this method. So in the future, this method can increase the scientificity, specificity, and enlarge its field of microbial species that can be detected. That's all my presentation. Thank you.
Okay, that's all from our presentation. Thank you for listening, and we are sorry if there are mistakes during our presentation. We close this presentation with Karma Santi. Om Santi 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 Om.